information that was transmitted had to be read by a manufacturing body within the cell, which puts amino acids together in a specific sequence, ultimately synthesizing a protein. This became known as the central dogma of molecular biology. Specifically, DNA serves as a template for the direct synthesis of a messenger RNA molecule, also known as mRNA, in a process known as transcription. Secondly, mRNA is read at a ribosome by transfer RNAs, also known as tRNAs, which work together to assemble a specific chain of amino acids, which collectively assemble to generate a protein, in a process known as translation. Proteins are the cell's internal machinery. Similar to parts of a car, each protein has a specific three-dimensional shape that determines its function. Any change in the shape potentially changes the function of the protein. Consider an analogy of transcription and translation to printing an essay from a computer. Once your essay is complete, you store the document to the hard drive. Similar to DNA, the hard drive stores information. DNA is a genetic storage molecule. Data in a computer is stored in a binary language. When it's time to print your essay, you send a command to the computer to send a message to the printer. This message is akin to mRNA, a genetic messaging molecule. Similar to the binary message sent to the printer, DNA and RNA share a chemical language based on nucleotides, hence why the information exchange from DNA to RNA is called transcription, an exchange of information in the same language. Once the information is received at the printer, it is translated from a binary language into a different language, a language of ink. Analogously, mRNA is read by the ribosome and translated into the language of proteins, which are made up of amino acids. Thus, the process from RNA to proteins is known as translation, translating from the language of nucleotides to amino acids. The ribosome is akin to the printer, serving as a facility for the process of translation. The molecules that actually translate the mRNA at the ribosome are a different kind of RNA, transfer RNA or tRNA. In the process of translation, a tRNA reads the mRNA and links a specific amino acid to a growing protein. For your essay to represent your idea, the ink must be physically arranged in a specific manner. Any malfunctioning in the positioning of the letters would not convey the same idea. Similarly, proteins have a specific three-dimensional shape that determines their function. Any change in that shape can potentially alter its function. In our analogy, DNA is the stored file in the hard drive. mRNA is the message sent to the printer. The printer is the ribosome. The essay is the protein, and the letters represent the amino acids. DNA indirectly codes for proteins. DNA directly creates all of the intermediate players of transcription and translation. DNA's day-to-day function is the production of RNA molecules. Messenger RNA is directly generated by a specific segment of DNA. That segment of DNA is known as a gene. The mRNA travels to a ribosome, which is made up of a protein and another type of RNA, ribosomal RNA, or rRNA. At the ribosome, the mRNA serves as a code for the synthesis of protein by linking specific amino acids in an exact sequence. The overall collection of an amino acid chain is the protein. DNA is also capable of self-replication, necessary for the creation of new cells. DNA and RNA are biological molecules known as nucleic acids. Nucleic acids, as well as proteins, are polymers, or molecules made up of a linking chain of repeating molecules. The repeating components are known as monomers. The monomers of nucleic acids are nucleotides, which are composed of three components: a sugar, a phosphate group, and a nitrogenous base. DNA, or deoxyribonucleic acid, is a double-stranded nucleic acid composed of monomers known as deoxyribonucleotides. A deoxyribonucleotide is made up of three components: a phosphate group, the sugar deoxyribose, and one of four nitrogenous bases. While the phosphate group and deoxyribose are identical in the varying deoxyribonucleotides, DNA houses four different nitrogenous bases: adenine, known as A, thymine, T, cytosine, C, and guanine, G. These four different nucleotides serve as the letters of the genetic informational storage, which are transcribed into mRNA and eventually read at the ribosome to create a protein. All of the biological diversity on Earth in the world is based on the language of life, which only has 4 letters. These nitrogenous bases can be placed into categories based on their shape. Thymine and cytosine are each composed of a single carbon ring skeleton and are known as pyrimidines, whereas adenine and guanine are composed of 2 carbon ring skeletons connected together, one 6-sided and the other one 5-sided, and these are known as purines. Ribonucleic acids, RNAs, are single-stranded nucleic acid polymers made up of the monomers ribonucleotides. Ribonucleotides are identical to deoxyribonucleotides with 2 exceptions. First, ribonucleotides are made up of the sugar ribose, which has a hydroxide at the 2' carbon, whereas deoxyribose has a hydrogen atom at that location. The carbons of ribose and deoxyribose are notated moving clockwise from the oxygen ring, 1' to 5'. The 1' carbon connects to the nitrogenous base. The atoms attached to the 2' carbon differ between ribose and deoxyribose. Clockwise from the 2' carbon is the 3' carbon, followed by the 4' carbon, and the 5' carbon attaches to the phosphate group. So, relative to RNA sugar ribose, DNA sugar deoxyribose lacks an oxygen at the 2' carbon, hence deoxyribose. That single difference allows cells to differentiate between those 2 nucleotides. Second, ribonucleotides differ in their suite of nitrogenous bases. 3 ribonucleotides have the same nitrogenous base as the deoxyribonucleotides: cytosine, guanine, and adenine, while the 4th ribonucleotide is composed of the nitrogenous base uracil. Uracil is very similar to thymine except that there is a hydrogen atom at the 3' location of uracil, while thymine has a methyl or CH3 group there. The phosphate group is identical for both ribonucleotides and deoxyribonucleotides. Nucleotides link together in long chains to form a nucleic acid. Individual nucleotides are connected by a covalent bond that forms between the 3' carbon of the sugar molecule of one nucleotide and a phosphorus of the phosphate group of an adjacent nucleotide. In this reaction, a hydrogen atom is removed from the 3' carbon and hydroxyl is removed from the phosphate. These byproducts combine, forming water, in a reaction known as a condensation reaction. Following this reaction, the 2 nucleotides are connected by a phosphodiester bond, in which a phosphate group is linked to the 5' carbon of its original nucleotide and the 3' carbon of an adjacent nucleotide. Adding a 3rd nucleotide, the nucleic acid begins to take shape. In this developing nucleic acid, a phosphate is attached to a sugar, which is attached to a phosphate, attached to a sugar, and so on. A phosphodiester linkage involves 2 of the 3 components of a nucleotide, a phosphate and a sugar. Hanging off to the side of the nucleic backbone are the nitrogenous bases. This repeating pattern forms the backbone of nucleic acids. One end of a nucleic acid strand is bound by a phosphate group, while the opposite end is bound by a sugar, giving DNA and RNA directionality. The phosphate group terminus of a nucleic acid is referred to as the 5' end of the strand, as the 5' carbon is the closest carbon to the end of that molecule. The opposite end of the nucleic backbone contains a sugar terminus called the 3' end. The sequence of nucleotides in a nucleic acid is known as its primary structure. Scientists have standardized the notation of nucleic acid's primary structure by listing the nucleotides from the 5' end to the 3' end, 5' to 3'. For example, a segment of RNA, adenine, guanine, uracil, adenine, cytosine, would be notated A, G, G, U, A, C. RNA, or ribonucleic acid, is a single-stranded nucleic acid composed of ribonucleotides. The nucleic backbone of RNA is bound by a phosphate group at the 5' terminus and ribose, a sugar, on the 3' terminus. Each ribonucleotide has one of 4 nitrogenous bases: adenine, uracil, cytosine, and guanine. RNA molecules are predominantly responsible for actively synthesizing proteins. DNA synthesizes messenger RNA, mRNA, which transmits genetic information from DNA to a ribosome. The primary sequence of the mRNA determines the sequence of amino acid in the resultant protein. Ribosomes are hybrid complexes made up of proteins and a different type of RNA, ribosomal RNA, rRNA. At the ribosome, the mRNA is read and decoded by a third RNA, transfer RNA, or tRNA. In eukaryotes, a different RNA, small nuclear RNA, snRNA, is involved in modifying the mRNA after transcription and before translation. DNA is a double-stranded nucleic acid composed of deoxyribonucleotides, which vary from ribonucleotides by having a different 5-carbon sugar called deoxyribose. In living organisms, there are 4 deoxyribonucleotides that vary in their nitrogenous bases: adenine, thymine, cytosine, and guanine. Once the primary structure of DNA was solidified, the next question was, how are the nucleotides arranged to create the DNA molecule, or the secondary structure of DNA? The discovery of DNA's secondary structure was one of the most important biological discoveries of the 20th century. One of the first clues came from analyses conducted in the early 1950s by Erwin Chargaff, comparing the relative abundances of deoxyribonucleotides across a variety of organisms. Chargaff discovered that the relative abundance of guanine equals cytosine, and the relative abundance of adenine equals thymine. And what is most interesting about this is that he found this relationship across many different species of organisms. Chargaff's discovery was instrumental to scientists uncovering DNA's secondary structure. James Watson and Francis Crick suggested Chargaff's evidence strongly supports base pairing in DNA, in which deoxyribonucleotides of adenine attach to thymine, and guanine attaches to cytosine. In addition, Watson and Crick hypothesized that base pairing of deoxyribonucleotides suggests that DNA was most likely double-stranded. To acquire evidence of the actual molecular shape of DNA, Rosalind Franklin and Maurice Wilkins bombarded DNA with X-rays and analyzed how the radiation scattered, a technique known as X-ray chromatography. Analysis of the scatter plots from this technique allowed them to measure the distance between atoms in DNA, and they were able to conclude 3 things. 1. DNA has a consistent width. 2. Within DNA is a repeating pattern. And 3. The molecule must be helical. In collaboration with Franklin and Wilkins, Watson and Crick used the measurements to define the geometry of the components of the deoxyribonucleotides. Creating physical models of the nucleotides, literally paper cutouts, Watson and Crick tinkered with the different arrangements of the nucleotides to explain 1. Chargaff's rule, 2. A consistent width, 3. The repeating pattern of the nucleotides, and 4. The helical shape of the DNA. By arranging base pairing nucleotides A with T and C with G side by side in strands running in opposite directions, all of the discoveries could be explained. In other words, Watson and Crick suggested that DNA is composed of 2 strands, one running 5' to 3', connected to a second strand running 3' to 5'. This orientation is called anti-parallel. The nucleic backbone is composed of alternating phosphate and deoxyribose sugar molecules, with a phosphate on the 5' end of the strand and a deoxyribose on the 3' end. The strands twist to form a double helix, a spiral bounded on the outside by 2 nucleic backbones running in opposite directions, with the nitrogenous bases facing inward. Based on Chargaff's findings, Watson and Crick determined that the nitrogenous bases from adjacent DNA strands connect according to base pairing. The discovery of a consistent width of the DNA also supported the AT and CG base pairing. While adenine and guanine are different molecules, they're both purines and approximately the same size and shape. The same is also true of the pyrimidines, cytosine and thymine. However, purines consist of a figure-8 structure which is larger than the circular structure of the pyrimidines. For the width of DNA to be consistent with the variety of shapes found in the nitrogenous bases, Purines must connect with pyrimidines. A purine-purine base pairing creates a larger molecule width than observed, and a pyrimidine-pyrimidine base pairing would be too small. If a purine-pyrimidine base pairing explains the consistent width of a double-stranded DNA molecule, why does guanine, a purine, always appear to bind with cytosine, a pyrimidine, but never thymine, also a pyrimidine? Why doesn't adenine, a purine, bind with cytosine? With their physical models of the nucleotides, Watson and Crick deduced that the nitrogenous bases of the adjacent strands were held together by hydrogen bonding. Due to the differential in electronegativities, the hydrogens of the nitrogenous bases are partially positive, and the oxygens and nitrogens are partially negative. Hydrogen bonds form between the partially positive and partially negative atoms of adjacent nitrogenous bases. Investigating the shapes and interactions of these four nitrogenous bases, they discovered that guanine and cytosine were geometrically complements of each other and held together by 3 hydrogen bonds, while adenine and thymine are held together by 2 hydrogen bonds. Essentially, the A, T, and CG pairing are more stable than any other combination due to the complementarity of the molecular shape of the hydrogen bond orientation. All right, let's take a closer look at proteins. Proteins are biological molecules that serve as cellular machines in living organisms. These large molecules are specific three-dimensional structures involved in biological processes, such as cellular signaling, catalyzing chemical reactions, molecular transportations, as well as many other functions. Proteins are polymers consisting of long chains of monomers, amino acids. Of the more than 500 amino acids known, only 20 appear in proteins of living organisms. An amino acid is a relatively simple organic molecule. Attached to a central carbon by single covalent bonds are 1. A hydrogen atom, 2. An amine group, NH3+, 3. A carboxylic acid group, COO-, and 4. An R group, also known as a side chain. At around pH 7, as in water, the amine group of an amino acid attracts a proton, becoming NH3+, and acts as a base. The carboxyl group is negatively charged in water due to the high electronegativity of both oxygens pulling electrons from the hydrogen and losing the proton. Different amino acids vary in their R group. Of the protein-building amino acids, the R groups can vary in their size, shape, and polarity. Proteins being made up of chains of amino acids vary based on the interactions of the atoms within the amino acids and water. These interactions dictate the shape of the protein, which in turn determines its function. R groups vary in their polarity. Nonpolar molecules have relatively equal distribution of electrons via covalent bonding, while polar molecules have unequal distribution of electrons. The unequal distribution of electrons in polar molecules creates partially charged atoms. Polar R groups are hydrophilic, meaning they have an affinity for water due to the hydrogen bonds between the partial charges of the R group and water molecules. Nonpolar R groups are repelled by water or hydrophobic. Therefore, in a chain of amino acids, ones with the polar R groups will bend towards water and nonpolar R groups will bend away, affecting the eventual shape of a protein. Proteins are polymers of amino acids chained together by covalent bonds, known as peptide bonds. A peptide bond is a condensation reaction in which the oxygen ion of the carboxylic acid from one amino acid is removed, becoming carboxyl, and combines with 2 hydrogen atoms from the amine group of an adjacent amino acid to produce water. A covalent bond forms between the 2 amino acids when the carbon of the carboxyl group that lost the OH during the condensation reaction combines with the adjacent nitrogen of another amino acid that lost the hydrogen atoms, bonding 2 adjacent amino acids. This is a peptide bond. Amino acids link via peptide bonds, forming long-chained molecules, or polypeptides. Proteins have 4 levels of structure. The three-dimensional shape of a protein determines its function, and the shape of proteins are ultimately dependent upon the sequence of amino acids coded for by DNA. The unique amino acid sequence is a protein's primary structure. In humans, sickle-cell anemia is an inherited condition in which blood cells have a variant of the oxygen-binding protein hemoglobin. Sickle-cell anemia is considered a disease of the primary structure of proteins, as it is caused by the variation of a single amino acid in hemoglobin, a valine instead of a glutamate in the 6th position of 146 amino acid protein. While normal blood cells are rounded, humans with this variant produce sickled shaped red blood cells. Normal blood cells are elastic and flow freely through veins, but sickled red blood cells are rigid and tend to get stuck where the veins branch. This blockage starves downstream tissues of oxygen, resulting in a host of medical issues, including lower life expectancy. The body identifies the cells when they get stuck and destroys them. Healthy red blood cells typically live between 90 and 120 days, whereas sickled red blood cells have a 10 to 20 day lifespan. Therefore, people with sickle-cell anemia must produce much more blood, which is rich in iron, leading to an overall iron deficiency or anemia. Interestingly, sickle-cell anemia is an evolutionary advantage in certain circumstances. Prior to globalization, the highest rates of sickle-cell anemia occur in tropical Africa, the Middle East, and India, all malaria-dominated areas. Malaria is a single-celled eukaryotic parasite transmitted by mosquitoes that some consider the most deadly human disease ever. However, in sickled red blood cells, the malaria parasites cause the cell to rupture before they can successfully reproduce. Therefore, people with sickle-cell anemia have an evolutionary advantage over people with normal blood cells in these areas. In areas absent of malaria, sickled red blood cells are highly disadvantageous due to the host of medical conditions associated with this variant. All of this is caused by a single different amino acid in one protein, an alteration of the protein's primary structure. When amino acids are grouped into polypeptide chains, neighboring amino acids can interact via hydrogen bonding. These interactions can form a regular pattern which increases the molecular stability of the polypeptide chain. These patterns are known as the protein's secondary structure and form either corkscrew-shaped structures known as alpha helices or folded ribbon-shaped structures known as beta-plated sheets. Recall, oxygen has a high electronegativity while hydrogen has low electronegativity. This differential in electronegativity results in hydrogen bonding between neighboring amino acids. Within a polypeptide backbone, the hydrogen bonds can occur between amine groups and carboxyls. The partially negative oxygen of the carboxyl can bind with the hydrogen of the amine groups of other amino acids. In alpha helices and beta-plated sheets, hydrogen bonding occurs between amine and carboxyl groups of different amino acids. How the hydrogen bonding occurs between amino acids determines which of the two shapes emerges. Within a single amino acid of an alpha helix polypeptide chain, the hydrogens of the amine groups face the opposite directions relative to the oxygens of the carboxyl groups. Down the entire alpha helix, the hydrogens of the amine group face the same direction, and oxygens of the carboxyl group orient in the opposite direction. Within the alpha helix, a hydrogen bond forms when an oxygen of the carboxyl faces the hydrogen of a different amino acid further down the polypeptide chain. This accumulation of multiple hydrogen bonds stabilizes the polypeptide chain. Beta-plated sheets are also formed by hydrogen bonding between the amine and carboxyl groups of different amino acids. However, the orientation of these groups differs. In a single amino acid of a beta-plated sheet, the oxygen of the carboxyl and the hydrogen of the amine group face in the same direction. In the adjacent amino acid, the oxygen and hydrogen both face the opposite direction relative to the first. In beta-plated sheets, hydrogen bonds also occur between the carboxyl oxygen and the amine hydrogen between neighboring amino acids. However, the orientation of these atoms causes the structures to bend in a folded ribbon shape or beta-plated sheet. While the secondary structure of proteins is determined by the interactions between the amine groups and the carboxyl groups of neighboring acids, tertiary structure is defined by how the R groups of neighboring amino acids interact. These interactions result in a very specific folding patterns, eventually helping to stabilize the specific three-dimensional structure of the polypeptide. Several types of interactions occur between neighboring R groups. While the hydrogen bonding determines the secondary structure of proteins, hydrogen bonding can also occur between the R groups of a polypeptide chain. The 20 R groups of amino acids are either polar or nonpolar. Polar R groups have oxygen or nitrogen atoms, which characteristically have high electronegativity due to their high affinity for electrons. These polar R groups tend to bond with hydrogen atoms of neighboring nonpolar R groups or the hydrogen of the amine group of the peptide backbone. Nonpolar R groups can also form hydrogen bonds with the peptide backbone, either by interacting with the oxygen of the carboxyl group or the nitrogen of the amine group. While hydrogen bonding is relatively weak, the overarching abundance of these interactions forms very stable polypeptide structures. In living organisms, proteins are surrounded by water. Polar R groups are hydrophilic and bend to turn towards water, whereas nonpolar R groups are hydrophobic and turn away from water. Hydrophobic R groups tend to amass in the internal section of a protein, forming globular masses. While hydrogen bonding is facilitated by the interactions of partial charges, certain R groups have full charges and are involved in ionic bonding. This happens when completely positive R groups form ionic bonds with neighboring R groups that are completely negative. The overall structure of a fully functional protein is known as the quaternary structure. Most proteins are composed of several polypeptides. A polypeptide is composed of either a series of alpha helices with tertiary level interactions or a series of beta-plated sheets with tertiary level interactions. In the next lecture, you're going to learn about the genetic code and the specifics of transcription and translation.